Hello. Thank you. Yeah, so thank you all. My name is Manuel Copas. I'm a senior lecturer in genomics at the University of Westminster. I'm also a fellow of the Alan Turing Institute, which is the national AI capability in the UK. And I also have a company called GenoInsight, which is basically around health equity. My main interest as a native Spanish speaker has been throughout many years now to bring all the wonderful things that are being developed here in the UK and making them accessible to the Spanish-speaking world. So let's see if this works. Right, so let me start with that number. I mean, you've heard it already. 80%, right? Some people say 85%. It doesn't matter. The point is that the vast majority of the data that we have is basically using, as you know, European ancestry data. So what that means really is that it means that South Asia is underrepresented. It means that Latin America is underrepresented. It means that Africa is underrepresented. I've not done anything there. It means that indigenous nations are underrepresented. And this is not an ethical issue, actually. It's a scientific problem, because this imbalance propagates through everything that we do. Genome-wide association studies, polygenic risk scores, drug target discovery, and increasingly, AI models trained on genomic data. So the implication of this is that we are building precision medicine on an incomplete foundation. And incomplete foundations produce unreliable results. Next slide, please. So let's see if, do you mind helping me with the slides? Oh, okay. So here's something that surprises many people. So genetic diversity does not follow census size. Look at Latin America, which is the place where I'm working the most right now. It's around 8% of the world's population, but 16% of known global genetic diversity is from Latin America. That's double the representation you would expect. Africa, 17% of the global population and around 25% of the known diversity. And I emphasize known diversity, okay? Because there's a lot of unknown diversity as of today. And Asia, 45% of the world's population, but less than 20% of known diversity. This gap tells you something important. Asia isn't less diverse. Asia is less studied. Indonesia alone has over, according to my statistics, 1,300 ethnic groups. I suppose the number changes depending on how you count it, but certainly it's one of the richest sources of human genetic diversity on Earth. And yet, Indonesia remains largely invisible in the databases that drive genomic science. So this isn't a data gap, it's a scientific blind spot. I don't know where to point this at. Okay, there you go. So why this matters for Indonesia? Well, and this partnership between the UK and Indonesia. Indonesia is extraordinary. Over 1,300 ethnic groups and thousands of years of population history. So it's a genomic resource that could transform our understanding of human biology. But here's the challenge. Polygenic risk scores developed in European populations, they don't transfer cleanly. The predictive accuracy drops. What works in London may not work in Jakarta. And there's another issue, sovereignty. For too long, global genomics has operated on an extractive model. Data flows out, benefits don't flow back. That approach erodes trust, and it undermines equity. And frankly, it's not sustainable. So both Indonesia and Latin America face the same structural challenge. Both are diversity rich, but data poor. I know this because of my own research, focusing on the Peruvian Genome Project, and the parallels are striking. This is where the UK-Indonesia collaboration can lead, not by extracting data, but by building together. So, what do we need to build? I see 3 pillars. First, equity-aware biobanks. What this means is representative sampling, not convenience sampling. It means community governance, communities involved in decision about their own data. It means local benefit and research that serves the populations it studies. Second, we need mandatory diversity, auditing for genomic tools and AI models, cross-population validation before deployment, transparent tracking of data flows. If a tool hasn't been validated in Indonesian populations, we should know that. Third, equitable partnerships. That means co-ownership, not extraction. Capacity building, not just data collection. Shared governance, not one-sided control. These aren't aspirations. These are engineering requirements. Representation must become measurable, enforceable, and architected into the system. Let me close with 3 points. First, genomic diversity is scientific infrastructure, not ethical decoration. It's not something that we do to be nice. It's something we do because our science depends on it. Second, Indonesia and Latin America are partners in this work, not data sources to extract from. That shift in framing changes everything about how we design collaborations. Third, precision medicine cannot be precise with partial data. The UK-Indonesia partnership, represented in this room, has an opportunity, and this opportunity that we have allow us to build genomics infrastructure that works for everyone, not just for the populations that have historically been overrepresented, but also for the underrepresented. Let us build the partnerships that make equitable genomics real. Thank you.